Hello everyone. In today's video, we will be learning about downstream processing in industrial microbiology. In this video, we will concentrate on removal of soluble products or cells from a culture broth. In industrial microbiology, the production process involves two steps. The first step is called as upstream processing and the second step is called as downstream processing. Upstream process involves inoculum preparation, media formulation, fermentation condition and fermentation process. In theory class, we have almost dealt with all the topics in upstream processing, whereas downstream processing involves the steps that has to be carried after the fermentation process is complete. Downstream processing refers to the recovery and purification of biosynthetic products after fermentation is complete. Now, let us see the steps that is involved in downstream processing. Microbial products usually is of two types. The first type is called as intracellular products and the second type is called as extracellular products. Intracellular products means the product that is produced during fermentation will be present inside the cell. Extracellular product means the product will be released into the culture broth. Based on the product, if it is an intracellular product or if it is an extracellular product, based on the product type, the steps in downstream processing will slightly vary. Say if the product is intracellular, then first the cell has to be lysed. So cell disruption has to be carried out and then we should go for removal of the product from the culture broth. If it is an extracellular product, then we have to directly remove the cells and the product will be present in the culture broth. So in this way, the steps will slightly vary based on whether it is an intracellular product or it is an extracellular product. For cell disruption, we carry out various methods like chemical methods, physical methods and enzymatic methods. After cell disruption, the insoluble products and the cells will be removed by various methods like centrifugation, flocculation, filtration and sedimentation. After the cells and or the product is being removed, the solid is being separated from the liquid, we go for the next step that is called as isolation or it is also called as concentration. In this step, we are going to remove the excess water molecules that has been present in the culture broth. Therefore, we are concentrating the product here. These steps involves evaporation, extraction, precipitation and membrane filtration methods. After the product is being concentrated, the next step is purification. During the product has to be purified. To carry out purification, the most frequently used technique is centrifugation. After the product is being purified, the last step is called as finishing step or it is also termed as formulation. Once finishing is done, the product that is being present cannot be sold directly. So therefore it has to be formulated. It has to be dried, powdered or crystallized and then it has to be sterilized and then only that product will be called as a final product. So before the product is being sold, it has to be formulated. So these are the various steps that is involved in downstream processing. In today's video, we will be only concentrating on removal of insolubles from the culture broth or removal of cell biomass from the culture broth. So in this step, the product recovery is the separation of whole cells or cell biomass or other insoluble products from the culture broth. Separation of cells or products from the fermented broth involves various methods and these methods are filtration, centrifugation, sedimentation, flocculation and flotation. Now first, we will concentrate on the first method, that is filtration. Filtration is a mechanical separation method. Filtration is the most commonly used technique for separating the biomass and culture filtrate. The efficiency of filtration depends on many factors: the size of the organism, presence of other organisms, viscosity of the media, and temperature. Several filters are used, such as depth filters, absolute filters, rotary drum vacuum filters, and membrane filters. We will now learn what are the different types of filtration methods that will be used. Now, you can see in this diagram, there is a membrane that is being present. Through this porous membrane, the culture broth will be separated. So when you put a culture broth into the on the porous membrane, what happens is, in the porous membrane the cells or the products will be trapped and the filtrate will be sent out. Now, the first type of filtration is called as depth filters. These depth filters are composed of filamentous matrix such as glass wool, asbestos, filter paper. The particles that are trapped within the matrix and the fluid passes out. Filamentous fungi is usually removed by this method. The next type is called as absolute filters. Absolute filters are having membranes with specific pore size. You should know that depth filter do not have a specific pore size because the matrix that we are using is glass wool, asbestos, and filter paper, where the pore size is not mentioned. Whereas on absolute filters, the pore size will be mentioned. There are various ranges of pore size available. The membrane has pores, and the sizes of this pore sizes specific based on the culture that we are using. For a bacteria, we use a different absolute filter, and for fungi we use a different absolute filter because of the cell size. So these filters has a specific pore size that are smaller than the particles that has to be removed. The next type is rotary drum vacuum filters. These filters are frequently used for separation of broth containing 10 to 40% of solids and particles in the size of 0.5 to 10 micrometers. Rotary drum vacuum filters have been successfully used for filtration of yeast cells and filamentous fungi. The equipment is simple with low power consumption and it is very easy to operate. As you can see in this image, the filtration unit consists of a rotating drum, and that rotating drum is partially immersed in a tank of broth. As the drum is going to rotate, it picks up the biomass which gets deposited on the drum in the form of a cake. This filter cake can be removed very easily using a scraper. Now, the next type of filtration is membrane filters. In this type of filtration, membranes with specific pore size can be used. However, this membrane filtration method has a limitation that the membrane usually gets clogged. So, therefore, the membrane has to be cleaned every now and then. And here, the vacuum is used to filter the culture broth. That is the speciality of membrane filters. Now, membrane filtration method can be carried out in two steps. The first step is called as static filtration and the second step is called as cross flow filtration. In static filtration, it is like our usual filtration method where the membrane will be placed in a culture tank and the culture solution will be poured into this. So the membrane is going to hold all the cell biomass and the filtrate will be liberated, which can be collected. In cross flow filtration, the culture broth is pumped in a crosswise fashion across the membrane. This reduces the clogging of the pores and hence better than the static filtration. Here, because of the cross flow, the cells do not settle into the membrane. So therefore, clogging of the membrane can be avoided. Now, the next step is centrifugation. After filtration, the another method that we can use for removal of substances or insoluble materials is the centrifugation method. Here we are going to use a centrifugal force. The technique of centrifugation is based on the principle of density differences. between the particles to be separated and the media. We know that when the centrifugal force is applied, the heavier particles is going to settle down faster, forming a pellet, and the broth will form a supernatant. The liquid part is going to form a supernatant and the solid part is going to settle down. Thus, centrifugation is mostly used for separating solid particles from liquid phase. Now, we will move on to the different types of centrifugation. The Usually in industries, the centrifuge that we use in the lab is different, and the centrifuge that we use in the industries will be different, because the capacity should be used, because in industries the product that is formed high is too much. That is why the type of centrifuge that we use will also differ. The first type is tubular bowel centrifuge. This is a simple and a small centrifuge commonly used in pilot plants. Pilot plants means before moving on to the huge fermenter tank, we are going to carry out our experiment in a small fermenter tank. So in such cases, this tubular bowel centrifuge will be used. Tubular bowel centrifuge can be operated at high centrifugal speed and can be run in both batch and continuous mode. In this, the cells are usually, the solids and the cells are removed manually. So here if you see this diagram, if you see here, there is a tubular like centrifuge. Now in this machine you are going to put the culture broth inside through this, and the tubular bowl is being rotated. So when once it is being rotated, the fluid is going to move out and the cells are going to settle at the bottom. So the cells can be removed through tubular bowl centrifuge. The next type is called as disc centrifuge. Now this disc centrifuge is similar to tubular centrifuge, whereas in the disc centrifuge there are various plate like materials that is being attached. It consists of several discs that separate the bowl into settling zones. The feed or the slurry is fed through a central tube. Now, during this, a centrifugal force is applied where the cells will be settled at the bottom and the clarified liquid can be sent out. So, that is about disc centrifuge. Moving on to the next type of centrifuge, that is multi-chamber centrifuge. In here, this is also similar to tubular centrifuge, whereas the centrifuge has many chambers. So here the fluid flows in and it will pass through different chambers with a high centrifugal force, and the clarified liquid will be removed and the cells will be settled at the bottom. This is about multi-chamber centrifuge. Then you have scroll centrifuge or decanter. This is basically a modification of tubular bowl type of centrifuge. It consists of several chambers connected in such a way that the feed flows in a zigzag fashion. There is a variation in the centrifugal force in different chambers. The force is much higher in the periphery chambers. As result, smallest particles settle down in the outermost chamber and the heavier particles will settle in the innermost chamber. The cells will be separated out and the liquid will be separated out in this scroll centrifuge or decanter. The next method is to remove the solid or the cells is flotation. Now you can imagine that when you take a glass beaker and you add water and soil into that. So let us allow the soil to settle down. So once you start mixing using a glass rod, you start mixing the beaker inside the beaker. What happens is the soil particles will move upside. The same principle is used in flotation. When a gas is introduced into the liquid broth, it is going to form bubbles. Because these bubbles are formed, these bubbles will join with the solid particles. The cells and other soil particles will get absorbed on gas bubbles, and these bubbles are going to rise up. When they rise up, they are being collected. The next method is flocculation. In flocculation, the cells or cell debris from large aggregates, they are going to form huge aggregates and they will settle down and they can be easily removed. So here we are going to use certain flocculating agents for allowing the cells to flocculate. Here the cells is going to join together. To make them join, we have to use certain flocculating agents. Now these flocculating agents are inorganic salt, organic polyelectrolyte, or some mineral hydrocolloidal substances. So when you add these substances, what happens is the cells are going to aggregate together. Once they aggregate together, their size will increase and the cells will become heavier, therefore they will settle down. And the last step is sedimentation. See, the sedimentation can be carried out by just keeping the broth for several days as such. So what happens is the cells, because it is heavy, it will settle down. But if you add a flocculating agents, the cells will aggregate and they sediment faster. They are going to settle at the bottom, and that process is called as sedimentation. Once all the particles are going to sediment, the broth that is present, the liquid part will be on the upper surface, so that will be decanted and the cells can be removed. So using these various methods, we can remove the cells or other products from the culture broth. These are the various methods used in industries for removing the insoluble materials or cells from the culture broth. This step is also called as separation of solids from liquids. Thank you.